All right, let's go through paper 4, which is the extended theory paper for biology. This is IGCSE, came from Cambridge International Examination, CIE, May-June 2017. And this is variant 2, so 0610/42. All right, let's begin.
Let's begin. Wait. What?
Question 1. Wheat is a crop plant grown to produce flour. A. Flour is used to make dough for bread. 1. State the name of the organism used to make dough rise. Okay, so the organism is yeast. If you want to be less specific, but you'll still get the mark, you could say a fungus, because yeast is a fungus. Or you could be more specific and call it Saccharomyces cerevisiae. But if you don't have to remember that, because Saccharomyces cerevisiae is the genus and species. All right. 2. State the name of the process that this organism uses to produce the gas that makes dough rise, and that is called respiration. Respiration. All right, the carbon dioxide bubbles are what make it rise. B. Farmers grow crop plants such as wheat to provide food. 1. State 2 types of natural events that could cause widespread food shortages. Keywords there, we have natural events, so nothing man-made like acid rain or anything like that, and widespread, so it has to be something that's going to affect more than just one farmer's field. So a tornado is not a widespread thing, it just does large amounts of damage but in a small area. Big widespread problems can be droughts. It can also be flooding. So the exact opposite of droughts, you can say flooding. Okay, but there's not just 2 things you can choose from. There's so many things. You could say desertification, so a large area becomes a desert. That can happen from a few different things, droughts and cutting down trees and that sort of thing. A tsunami, that can cause widespread food shortages. Monsoons, they affect quite a large area. Hurricanes can affect large areas. Cyclones can affect large areas. Other things you can say, earthquakes, volcanic eruptions. We could talk about a specific disease, say a disease of a particular crop plant. So you could say pretty much anything that is a natural event that affects a large area. All right. 2. The increasing human population is another reason for food shortages. So these are not natural events. These are to do with humans. Explain how the increasing human population contributes to food shortages. All right, so this is a 3-mark question. Again, there are lots and lots of things you can choose from, but they have to be something related to increasing human population. So one of the things you could say is that because we have more cities, there is less land for farming food available. So another way to say that is limited land for farming because of increased urbanization. Another thing that can contribute to food shortages is the fact that not all farmers grow food. They can grow other cash crops such as biofuels or tobacco. Some land is used for cash crops such as biofuels or tobacco instead of food crops. Okay, another thing you could put down is you could say that there's quite a lot of food spoilage and wastage because there's an unequal global distribution of food. So there's a lot of food spoilage and food wastage due to an unequal global distribution of food. Other things you could say is that basically because there's more people, there's an increased demand for food. More people means we need more food to survive. Areas that have war or a lot of poverty are much more likely to have food shortages. Another thing that can contribute to food shortages is poor farming practices. So some farmers don't do crop rotation, which means that the land ends up getting depleted of many nutrients, such as nitrogen. Pollution. So more people means more pollution. So water pollution or air pollution, acid rain, they can all be linked to crop failure. You can say about eutrophication, because that can affect the water quality, which can affect the crops. You could say that there's a lot of erosion where there's lots of land being used. And erosion means that the quality soil for growing good crops will get blown away or washed away. You could say that, because it's not just crops, there's also fishing. You could say that humans Also overfish. There's lots and lots of things you can put down. Don't write a whole essay. The examiner only wants to see 3 marks worth. 3 good marks. Make sure you're comfortable with them. Okay, next. C. Many farmers grow crop plants as monocultures to improve yields. Figure 1.1 shows a monoculture of wheat. So a monoculture, mono meaning one culture, one type of plant. If you look at this picture, as far as the eye can see, other than this big piece of machinery here, it is just wheat. That's all that's being grown in that area. Okay, the question is, describe the impact of large-scale monocultures on the environment. So this is a 4-mark question. So you have to have 4 good points again, but again, there's lots of things you can put down. One consequence of planting large-scale monocultures is that you lose a lot of the biodiversity. Well, all the biodiversity is pretty much just one plant. And because there's just one plant, it's very easy for diseases and pests to spread throughout a large area, affect the whole field. And a field the size that was shown in the picture above is very, very big. That would be a big problem to the farmer and to all the people who want to eat that food. So the loss of biodiversity can result in outbreaks of diseases or pests. So another impact of large-scale monocultures is that if you only have one type of plant, you have lost a lot of the habitats for a variety of organisms. You don't have any large trees that birds can nest in, for instance. So it can result in a loss of the variety of habitats. Another impact of large-scale monocultures is that the area is very rarely already. It didn't start out as just an open field with nothing in it. They often had to cut down trees or maybe drain areas that were too swampy. So you can say deforestation. So deforestation to make space for crops. And also, it's not all bad. Another thing you could write down is that because they're doing it in a large area, it's very efficient. So that means that there's less land required than if lots of small farmers were doing it on smaller scales. Efficient food production, so less land required. Some other things that you could talk about how some food chains have been disrupted. Give examples. You can talk about loss of nutrients in the soil. So either they have to let the field go fallow for a year or so. That just means grow however it wants. Or do some crop rotation. Maybe plant some clover in between the wheat so that the nitrogen can be replenished. Another thing that can happen is that because those plants are sucking up a lot of water, it can result in decreased water in the soil and desertification, turning it into a desert. It can also cause soil erosion. Okay, and another thing that can happen, that can be the result of large-scale monocultures on the environment, is that these crops are generally use a lot of pesticides, and pesticides they can move with the wind. They can spread to other areas, affect the insects around it, but also some insects don't know that the boundary of the field is where they're not allowed to go. So, say butterflies that fly through the area will land on the crops and be affected. Okay, so there's lots of things you can write down. Again, make sure you have 4 good points in there. Question 2. Many researchers are studying the structure and function of genes. A. Define the term gene. This is one of the definitions you really need to know. So a gene is a length of DNA that codes for a protein. 2 marks there. One saying it's a length of DNA. It's not a strand of DNA, because a strand of DNA, you can be talking about a chromosome, the whole thing. And the other mark is saying it codes for a protein. It doesn't just create a hereditary trait. The hereditary trait is caused by a protein. Genes code for proteins. That's why the traits occur. B. Every living cell is able to make proteins. The process begins in the nucleus. Describe how proteins are made in a cell. So this is a 4-mark question. However, you need to make sure you are clear that you've described the whole process. So you probably have to put down more than 4 points. You don't necessarily have to put down all the points. I'll put down all the points, but you still want to make sure you put enough points down, and it needs to be in the right order. Okay, so proteins are made by ribosomes. So proteins are made by ribosomes. And how does that happen? The mRNA is copied from the gene which stays in the nucleus. So the mRNA is copied from the gene as the DNA stays in the nucleus. So the DNA can't move, the gene is on the DNA, and it's a messenger RNA. You have to put the M in front because it's not just RNA. You have to be specific. So what does that mRNA do? It doesn't stay in the nucleus. It moves out of the nucleus and finds a ribosome. So mRNA moves from the nucleus to the cytoplasm and finds a ribosome. You can talk about the endoplasmic reticulum here as well, the rough endoplasmic reticulum with the ribosomes on. So the ribosomes can either be on the endoplasmic reticulum or they can be free in the cytoplasm. So once it finds the ribosome, the mRNA passes through it. So mRNA passes through the ribosome. And the ribosome basically reads the mRNA and assembles amino acids so that they're in the correct order to make a protein. So ribosomes read the order of the bases on the mRNA and assembles amino acids in the correct order to make a protein. And this process uses energy. So protein synthesis uses energy. You can also talk about what determines the order of the amino acids. It's based on the order of the bases on the DNA, and that affects what the order of the bases are on the mRNA. So the order of amino acids is determined by the order of the bases on the DNA, which determines the order of bases on the mRNA. So it goes DNA, then it goes mRNA. This is not part of the answer. This is just to be clear about what that said. mRNA, then it goes in a ribosome. It goes to the amino acids are put together in the correct order to make a protein. Okay, so that's basically what you're describing here. All right, good. But you only need 4 marks. You do need to explain what's happening though. So it's difficult to get all that information really into 4 marks. I'm not sure how they expect you to do that. Really, it's okay to put more down. Just make sure you know what you're talking about. C. Figure 2.1 is a diagram of a protein used to move ions across the membranes in root hair cells. So root hair cells are on the roots, and it moves ions from low concentration to high concentration. So it moves it against the concentration gradient. Moves it against a concentration gradient, which means it needs energy. 1. State the name of the process that moves mineral ions into root hair cells through cell membrane proteins. Well, as it needs energy, it's active transport. 2. Explain how protein molecules move ions across a membrane during this process. All right. Okay, so what happens is you have the ions, low concentration, so not many, plus, minus, whatever they are, positive ions, negative ions, it doesn't matter, and they will attach to a spot on the protein. They will interact with the protein, change it slightly so that the protein opens a channel, or pulls it across basically, of the cell membrane so it can go into the high concentration. It's basically what happens, but it needs energy to do this. So the protein uses energy from respiration, and with that energy it can interact with the ion to change it so that the protein can change shape. The protein interacts with the ion to change the shape of the protein, and by changing the shape, that can mean that the ion will move against the concentration gradient. So it moves ions against the concentration gradient. So there's another mark available for any other valid point. You can mention how proteins have a specific shape to match the ions. Okay, so one protein will transport one ion. D. Proteins are also found in the blood. State the names of 2 proteins found in the blood. Well, there's quite a few things you could put down here. 1, you could say enzymes. There's lots and lots of enzymes. Well, there's enzymes everywhere, really. And enzymes are proteins. Another thing you could say is fibrinogen. And if you studied your section on blood clotting, fibrinogen is a protein necessary for blood clotting. There's also a lot of plasma proteins that are just in the plasma. Hemoglobin is in the red blood cells, which are in the blood. They're part of the blood. And they're proteins. You can also say antibodies. Antibodies are proteins. You can name a specific hormone, such as insulin, glucagon, ADH, things like that. Those are all proteins. You can put down any of them, but only put down 2. That's all the examiner wants. 3. Heart rate is influenced by electrical impulses from the brain. A. State the name of the type of cell that conducts electrical impulses to the heart. Okay? The type of cell. Okay, you can say it's a motor neuron, that's a type of neuron, or you can say it's a neuron or a nerve cell. It's not specific. Okay, it's a type of cell. Some people call them the effector neurons, and that's also correct. B. The electrical activity of the heart can be recorded on an ECG, electrocardiograph. Figure 3.1 shows an ECG of 1 heartbeat. So this is the electrical impulse in a heartbeat. So this is 1 beat. Table 3.1 shows how the electrical activity during 1 heartbeat corresponds to the opening and closing of the valves in the heart. Let's look at table 3.1. Complete table 3.1 using the words open and closed. Let's do our quick diagram of a heart. This is how I do it, and it might not be beautiful, but it works. It's nice and easy to draw quickly in an exam. You draw a heart. Okay, you put a line down the middle and a line down the middle. Now we have 2 vessels going into, one going into each of these. These are the atria, and these are the ventricles. Okay, these two lines here represent where the blood goes in. It goes into the heart. I'm just going to draw it all as red rather than divide between which one's red and which one's blue. Okay, it goes into the heart, the 2 sides there. And we have 2 more vessels that are long and go down all the way into the ventricles. And the blood goes up and gets pushed up, and gets pushed up. All right. And we have valves here and here. Those are the AV valves, the atrioventricular valves between the atria and the ventricles. We have valves here and here. Those are the semilunar valves. Okay, you can't have both sets of valves open at the same time. So both AV valves will be open at the same time, both semilunar valves will be open at the same time, but you can't have the semilunar valves and AV valves open at the same time. All right, hopefully that makes it easier. I find it easier, but hopefully you find it easier. Okay, so at the beginning of the heartbeat at P, what happens is it causes the atria to contract. Why do the atria contract? To push the blood into the ventricles. So if the blood needs to go into the ventricles, that means the AV valve has to be open. So if the AV valve is open, the semilunar valves are closed. Now at Q, R, and S, that's a big spike because it has to push the blood all the way up through these long vessels here to go either all the way to the body or to the lungs. Okay, that's a big push. That means that the semilunar valves have to be open, which means that the AV valves have to be closed. And then at T, the atria and ventricles relax. Why do they relax? Because you're trying to bring blood into the heart again. Start it again. So that means the AV valves have to be open and the semilunar valves have to be closed. Okay, you can't have them both open or both closed at the same time. That's it. Good. 2. State the function of heart valves. Well, heart valves are there to prevent the blood from flowing backwards. Another way you can word that is to ensure one-way flow of blood through the heart. Both ways of wording that would be absolutely correct. C. Figure 3.2 shows the ECG of an athlete before and during exercise. So this is at rest, so before exercise, and this is during exercise. It's not after exercise, it's during exercise. All right, 1. Calculate the heart rate in beats per minute before exercise begins. Show your working and give your answer to the nearest whole number. Give your answer to the nearest whole number. That's something that's often missed for these things. What that means is you can count one heartbeat as basically looking at the same point on the ECG. Same, or you could look at the part that goes down. Okay, you don't have to figure out exactly where it stops perfectly. You can just count them as one heartbeat. So there's two methods of calculating this. One, you can say the average number of beats per minute, or you could say the length of one complete heartbeat. You can find the length of one complete heartbeat and multiply that by 60. All right. So I'm going to show you both methods. All right. So the first method, if we look at this, the average number of beats per minute, how many heartbeats are in 1 minute? Okay, so if we look for the number of heartbeats in 5 seconds, because that's when it begins, we could say there's 1, 2, 3, 4 beats in 5 seconds. 4 beats in 5 seconds. There are, if you divide 60 seconds divided by 5 seconds equals, that's 12. Okay, so 4 beats in 5 seconds, so 4 times 12 equals 48 beats per minute. Okay, so you could say that is 48 beats per minute. It's already in there. Okay, so that's one method of doing it. Nice and easy. The other method of doing this is you could say the length of one complete heartbeat. So one heartbeat, let's look at this one. That's a nice, if we look at this point down here, between here and here, that takes 1.4 seconds. So one beat takes 1.4 seconds. So how many seconds does it take one beat? So if you look at this, and for 1 second, if they've gone 0.8 of a beat, it takes 0.8 seconds per beat, and you multiply that times 60 seconds. All right, so that will also equal 48 beats per minute. Okay, both of those work well. I'd say the first way is probably easier, but they both work. 2. Using figure 3.2, describe how the electrical activity of the heart during exercise differs from the electrical activity before exercise begins. Okay, when it says using figure 3.2, using something, it means it wants you to quote data. It wants you to quote data using the units. Okay, so we're just describing the difference. We're not explaining it. Okay, so a few things you'll notice. You'll notice that as soon as exercise begins, the beats are closer together. The beats are faster. The rate is faster. You might also notice that the height is about the same. It doesn't change. And so that's 2 marks you could say there. And the third mark you can say, because it's a 3-mark question, is you can quote a data point. So you can say how many beats per minute there were to start with. You've already calculated that, 48. How many beats per minute are there after? Okay, let's do this. Let's do the same way. We could say there's 1, 2, 3, 4, 5, 6 between 5 and 10 seconds. There's 6 beats between 5 and 10 seconds. So if you have 6 beats in 5 seconds, 6 beats, multiply that by 12, 6 times 12, 72 beats per minute after or during exercise. Let's write that information down. So you can say the electrical activity during exercise increases, or the beats per minute during exercise increase. Same thing. But the question asks about electrical activity. You probably want to refer to electrical activity in the answer. Okay, now you can say, you could explain what you mean by that. Before exercise, there were 48 beats per minute, and during exercise, there were 72 beats per minute. Okay, make sure you don't just write 48 and 72. You have to put in the units. They always look for units for this. And you could say that there is no difference in the height of the peak. So no difference in the heights of the peaks before and during exercise. Okay, this final point is very similar to the first point. It's different, but it's another thing you can put down. So the waves were closer together during exercise. So you could say the electrical activity during exercise increases because the waves were closer together during exercise, and that would be 2 marks. And then quote a data point. All right, so that's 4 possible marks for a 3-mark answer. You should be fine. 3, describe how the ventilation of the lungs will change while the athlete exercises. When they're talking about ventilation, they're really talking about breathing. So that's a 2-mark question. They want to talk about the depth of the breaths and the rate of the breaths. That's what they're looking for. So the volume of breaths will increase during exercise, or the volume of ventilation will increase during exercise. They'll have deeper breaths. So ventilation rate will be faster. If it's a ventilation rate or breathing rate, same thing. Okay, there we go. That's 2 marks for a 2-mark answer. Question 4. A balanced diet is required to ensure healthy weight gain as children grow. Explain the term balanced diet. Again, this is another definition you should probably know. So a balanced diet contains all of the nutrients in the correct proportions to maintain health. Okay, so a balanced diet contains all the nutrients in the correct proportions to maintain health. That is actually a 3-mark answer. All the nutrients is 1, correct proportions is 2, purpose is to maintain health is 3. If you're uncertain of it, you can always write down a couple other things. You can list the types of nutrients. So the types of nutrients, we have proteins. We have lipids or fats. Doesn't matter which one you call them. We have carbohydrates. We have minerals and vitamins and water and fiber. Those are the nutrients that we consider as the ones that you need for proper health. Another thing you could say, if you're uncertain about if this first one is worth 3 marks, is you could say that this provides the appropriate energy requirements, and that there are different energy requirements according to the age of the person, the sex of the person, male or female, the lifestyle, are they active or do they just sit around playing on their phones all day? Are they pregnant? So all these things will require different amounts of nutrients. Okay, so those are, if you're uncertain if this is worth 3 marks, you can always write down a couple extras. Only write them down though if you know that they're correct. B, a doctor diagnosed a young child with marasmus. Describe the symptoms of marasmus. Okay, so marasmus, basically the child does not have enough body weight. So they have a low body weight. They haven't really grown very much. They could have lost weight. So they could have a low body weight due to the lack of growth or losing weight. Okay, another thing that they can have is diarrhea and vomiting. Unpleasant. So they can have diarrhea and vomiting. They're also likely to be very tired and prone to infections. So likely to be very tired and prone to infections. Okay, so tired, another way of saying that is fatigued. Prone to infections, they could be prone to diseases or dehydration. They could be irritable. You're likely to be irritable if you're having a lot of diarrhea, vomiting, and you're tired. They can be dizzy, weak. Their wounds don't heal very quickly. They can have muscle wasting, so they'll be very, very weak with the muscle-wise as well. And you can describe how their hair or skin or nails could look. So lots of things you could write down, but only write down 3 that you know are correct. C, the child with marasmus was put on a special diet. He was given fortified milk, which is milk that has extra nutrients added to it. The child was encouraged to drink as much fortified milk as he wanted over a period of 7 months. Table 4.1 shows the composition of the fortified milk given to the child and the composition of cow's milk for comparison. Okay, so if we look at this, fortified milk has a lot more protein than cow's milk. Also has a lot more carbohydrates than cow's milk and a lot more fat than cow's milk. And because it has all those things, it has a lot more energy than cow's milk. Okay, and that's probably a good thing. The body mass of the child who had marasmus and the mean body mass of the healthy children the same age were recorded, and the data is shown in figure 4.1. Okay, so this is figure 4.1. So the healthy child is on the top solid line, and the child with marasmus, that this is the point where he's given the fortified milk, and if you can see, the body weight increases a lot very quickly, and then it becomes about the same as a healthy child, which is a good thing. Using the information in Table 4.1 and Figure 4.1, describe and explain the importance of diet when treating children affected by marasmus. Now, this is a pretty big question. This is using the information in Table 4.1 and Figure 4.1. So that is data quote, data quote, describe and explain. So these two have to be linked together. All right, so this is a pretty big question here. This is a 6-mark question, but there's, again, lots and lots of things you can put in. So the first thing that's most obvious to see, I think, is that the child with marasmus has a much lower body mass than the child that's healthy. But once the child is given the fortified milk, as we said, this increases quite dramatically until it becomes much more like the healthy child. All right, well, it's only 2 children. There's going to be slight variations, which means that this trend is the same. All right, so why does it increase? It increases because the child was given the fortified milk as much as they wanted to drink, and in the fortified milk, they contain proteins that are required for new cells to grow, for muscles to grow, and for repair. It contained carbohydrates and fats that are needed for energy, and energy is needed for respiration. Fats are also used for insulation, for the cell membranes, for protecting organs, for neurons. Okay, so that's a lot of marks you can put in there. You could say that there was a rapid increase in growth in the body mass of the child with marasmus, and then what's this? This is at about 16 and a half months. This is 17 months. At about 16 and a half months, or 17 months, somewhere around there, the children have about the same mass. It's only 2 children, so it's not a huge sample size. Okay. You could say that the reason why the rapid growth as well was because the child with marasmus was encouraged to drink as much of the milk as possible. All right, and then you can quote some data. You can say, well, what was body mass? What was the difference in body mass at the beginning at 13 months, and compared to the body mass at the end at 20 months? Was it the same? And from the table, the data that I would quote from the table is the energy in both. Okay, the energy is very important. All right, so let's start writing some of that into a coherent answer that will get you some marks. So the child with marasmus has a lower body mass than the healthy child. There was an initial rapid increase in the mass of the child with marasmus because he was encouraged to drink as much of the fortified milk as he wanted. So here you're saying there was an initial rapid increase, and the reason because is the linking to explaining. Because he was encouraged to drink as much fortified milk as he wanted. So the fortified milk has more nutrients and energy than the cow's milk. Here's a good place to point to do your data quote from the table. The fortified milk contains 5,468 kilojoules per decimeter cubed, whereas the cow's milk only contains 2,845 kilojoules per decimeter cubed. Again, put in the units. So at 16.5 months, the trend was similar to the healthy child. Okay. So you can also mention that protein is required for muscle growth and repair, carbohydrates are required for energy, for respiration, and fats are required for energy as well, and for insulation. All right, you could also quote another data point from the graph saying that initially at 13 months, the body mass of the child with marasmus was 4 kilograms and the healthy child was 6.75 kilograms, but at 20 months, the child with marasmus was 10.5 kilograms and the healthy child was 10.3 kilograms. Okay, so that's another thing you can put down. So there's a lot of information you can put in. That's at least 6 marks. A couple of these points may have more than one mark in them. Okay? Don't be afraid to write in a little bit more than is necessary. You'd want a little bit more than a little bit less. But don't write a massive essay. Make it easy for the examiner to find the marks by writing your answers in bullet points. D. It is important that children with marasmus produce enough bile. Well, it's important for everybody to produce enough bile, but it's more difficult for children with marasmus. Describe the role of bile in the digestion of fats. Okay, bile is necessary for emulsification. So bile is necessary for the emulsification of fats. And remember, if you describe what emulsification is, it's not breaking the fats into smaller molecules, because that describes chemical digestion. It's breaking the fat into smaller droplets, and that's the mechanical digestion. So bile does mechanical digestion, not chemical digestion. So this increases the surface area for the lipase to digest the fats. So bile also neutralizes stomach acid to provide the optimum pH for lipase and speed up digestion. Another word for stomach acid is chyme. Okay, so that is the answer to that question. Question 5. Acid rain has negative effects on many ecosystems. A. Describe the negative effects of acid rain on freshwater ecosystems such as streams, rivers, and lakes. All right, so the most important thing that affects everything else is that acid rain decreases the pH of the lakes and rivers and streams to make them acidic. Acid rain decreases the pH of streams, rivers, and lakes, making them acidic. And that does a lot of things. One of the things it does is it makes aluminium ions and some other ions mobile in the water. So it makes aluminium ions mobile. So aluminium ions are found in the rocks quite often, and they're fine when they're in the rocks, but when they're just as ions, they're very toxic. So acid and aluminium ions are toxic to aquatic animals. Another thing that acid rain does is it damages shells. It damages shells of mollusks and of birds that nest near the water. So the acid damages shells of mollusks and birds that nest near the water. So carbon dioxide. The carbonate in the shells reacts with the acid to produce carbon dioxide, and it just dissolves the shells. Acid is also very hard on fish and frog eggs, not because of the calcium carbonate in the shell. They don't have shells, but it makes it very difficult for them to reproduce. So fish and frogs fail to reproduce. Another thing that happens is that aquatic plants become damaged, and many of them will die. So it damages aquatic plants and causes many to die. And there's obviously a lot of insects that live in the aquatic plants as well. And because of all this, there is a huge loss of biodiversity. There's a loss of biodiversity. Another thing that can happen is that fish will produce mucus to protect themselves against the acid, but this mucus blocks their gills so they can't absorb oxygen from the water, and they die. So fish produce mucus which block their gills. Okay, this is a lot of points, certainly more than the 5 marks. Other things you could put down, you could talk about other nutrients that are leached from the soil. You can describe a particular food chain that would be disrupted by acid in the lakes. You can do any other valid point, which you can talk about denatured enzymes, loss of habitat, if you're talking about maybe the plants. Okay, there's lots of things you could put down, but 5 marks is what you need. B. 2 groups of students were asked to investigate the effects of acid rain on the germination of pine tree seeds. 1. Both groups of students decided to use sulfuric acid rather than hydrochloric acid to represent acid in their investigation. Suggest why. Well, the reason why is because acid rain is often caused by sulfur dioxide and not by a derivative of hydrochloric acid. So acid rain is often caused by sulfur dioxide. 2. State how the students could have measured the pH of the sulfuric acid. There's a few ways. You could use a universal indicator solution or another pH indicator. You can use a pH meter, pH paper, pH probe, pH sensors. I'm going to say a pH meter. So they could use a pH meter. All right. You could write it in a sentence if you like. It doesn't matter. All right. Just one method is what they ask. 3. The students provided the environmental conditions that seeds need to germinate. State 2 of these environmental conditions. Okay, for germination, seeds need water, so moisture or humidity. They need warmth or heat, and they need oxygen. They also need a proper pH. So just put down 2 of those. So I'm going to say water and oxygen. They do not need light. Okay, most seeds don't need light in order to germinate. You can germinate them under the soil. You don't get light if you're planted inside the soil. C. One group of students recorded the change in temperature as the seeds germinated in 5 insulated flasks labeled A to E. 1. Suggest why germinating seeds increase in temperature. Well, germinating seeds are doing respiration, aerobic respiration actually, and this releases heat energy. So germinating seeds are doing aerobic respiration, which releases heat energy. 2. The students sterilized the seeds before their investigation. The teacher told them to use a sterilizing solution to wash the seeds because steam would kill the seeds and prevent the seeds from germinating. Explain why steam sterilization would prevent seed germination. Well, steam denatures enzymes. Steam is hot water, so it denatures enzymes. Steam denatures enzymes. Remember, it doesn't kill enzymes. Enzymes are not alive. The seed is alive, but the enzymes are not. They get denatured. They don't get killed. The results from this group of students are shown in table 5.1. So this is a table. We have a flask A to E. We have a pH, pH 2 to pH 7, acidic to neutral, and we have the temperature is about the same for the first 3, and then it goes up to 46 degrees. Pretty much over doubles between A, B, and C to E. All right, so describe the results obtained by group 1. Okay, it doesn't say use the table, but they're implying that you need to use a table, so that's a data quote. So the temperature increased as the pH increased. That you can see quite easily. It doesn't go beyond neutral. It's likely that if it went to pH 8, 9, 10, the temperature would start decreasing again. So then you'd have to say increased and then decreased. But here you just have to say it increased as a pH increase. That's all they looked at. Okay. Another thing that, as I said before, is that these 3 are all pretty much exactly the same. 0.3 degrees between these 2, 0.6 degrees. It's not that much of a difference. All right. So there was little difference at a pH of less than 4.0. Okay. Now, as we said, you can also quote a data point. At pH 7.0, the temperature of the seeds was 46.2 degrees Celsius, but at pH 2.0, the temperature was only 20.6 degrees Celsius, and that would work. D. The second group of students placed 100 seeds in each of 5 petri dishes labeled 1 to 5. Group 2 used the same range of pH values as Group 1. They recorded the percentage of seeds that had germinated after 72 hours. The results are shown in Table 5.2. Okay. So this is the second group of students doing exactly the same thing. The teacher thought that group 2 had forgotten to add the sulfuric acid to one of their petri dishes. Suggest which petri dish was not set up correctly. Well, look at this. We have 20 degrees, 82 degrees, 19 degrees, 65, and 87. This doesn't seem to fit, does it? Because pH 4 and under, these should be pretty much exactly the same. So we could say petri dish 2 at pH 3.5. All right, that's good. Question 6. Figure 6.1 is a flow diagram that shows what happens at the start of a bacterial infection. Okay, so we have our white blood cells here, and we have our bacterial infection, our bacteria, here. I'll label all these other parts in a moment. A. Cell R is a prokaryote and cell S is a lymphocyte. 1. State the names of 2 cellular structures that would be found in both prokaryotes and white blood cells. Okay, so we need things that are in both of them. One thing that they both have, a cellular structure, are cell membranes. Okay, and they both have DNA. They also both have ribosomes, and they both have cytoplasm. Okay, those are the cell structures that are in both of them. 2. Describe how the cellular structure of white blood cells differs from the cellular structure of prokaryotes. All right, so there's many things. Well, this one's only 3 marks, so we have to put down 3 different things, but there's a lot more than 3 things we can put down. All right, so we can talk about Cell walls. Well, white blood cells have no cell walls because they're animal cells, but prokaryotes do have a cell wall. They have the peptidoglycan cell wall, different to the plant cell walls. Okay, another thing you talk about are membrane-bound organelles. So white blood cells have organelles such as mitochondria, that's an example, or the nucleus, but prokaryotes have no membrane-bound organelles. Remember, a ribosome is not a membrane-bound organelle. It doesn't have a membrane around it. So that's why prokaryotes can have ribosomes. And we can also talk about the DNA. As I said, they both have DNA, but they're not the same. Well, it's the same bases, but the DNA is not the same structure, same shape. So white blood cells have linear DNA with proteins called histones associated with them to help them coil, but prokaryotes have loops of naked DNA, naked meaning they don't have proteins, and small plasmids. Okay, it's three different cellular structures. Other things you can talk about, you can say the white blood cell has a nucleus, but prokaryotes only have a nucleoid region, no nucleus. You could say that the ribosomes in white blood cells are large. They're 80S. That's just a size. You don't need to know that. And they're small in prokaryotes. They're 70S. They're smaller. You can also say that the white blood cells are much larger than the prokaryotes. And you can talk specifically about the white blood cells, not just a general eukaryotic cell as well. And white blood cells produce antibodies, but prokaryotes do not produce antibodies. Okay, so lots of things you can put down there. 3 marks. Make sure they're 3 marks you're very confident with, and write those ones down. B. Cell R is a pathogen that has structures T on its surface. These structures are recognized by cell S. Cell S is a lymphocyte and it produces structures V. Cell R reproduces by binary fission, and cell S divides by process U. Identify T to be from the passage and figure 6.1. It's probably easier to identify them from figure 6.1. So we need T, U, and V. Okay, so as I said, I would be explaining this one later. Now is the later. All right, so we have S. It said in the passage that S is a lymphocyte. R is a pathogen. Although up above it says it's bacteria. So it's a bacterial pathogen. Okay. T, now these are structures on the outside of the pathogen that identify it to the body as being not of this body, not self. So these are called antigens. Antigens. All right. Now the process U is it's a cell inside your body that is not one of your egg or your sperm. So it's any other cell that's not an egg or a sperm inside your body divides by mitosis. Mitosis makes mitose, and every other cell that's not an egg or sperm. Okay, meiosis makes only egg or sperm, but mitosis makes mitose. Okay, and V, now these are little Y-shaped proteins, making them bigger so they can see them a bit better. This Y-shaped protein will fit onto the antigen. These are called antibodies. So we need T for antigen, U for mitosis, and V for antibodies. All right. So we have antigens and mitosis makes mitose and antibodies. All right. C, cell W, and that's this big cell here. In figure 6.2 also responds to pathogens. So this is a pathogen right here. This is the pathogen. And cell W, it looks like it's eating the pathogen. 1. State the name of the process shown in figure 6.2. Well, the white blood cell that goes around eating up pathogens is called, the process is called phagocytosis. There we go. And 2, describe what happens to the pathogen during the process shown in figure 6.2. All right, so the pathogen is engulfed by the phagocyte. The phagocyte engulfs the pathogen. Okay, it engulfs the pathogen or surrounds the pathogen. It does not eat the pathogen. It doesn't have teeth. It doesn't have stomach acid. It's only one cell. Okay, so you have to be careful. You can't say the phagocyte eats the pathogen. It doesn't. Okay, so phagocyte engulfs the pathogen and it creates a vacuole surrounding it called a phagosome. And in this vacuole, the phagocyte puts enzymes in that break down the pathogen. So the phagocyte engulfs the pathogen and forms a vacuole around it. Enzymes break down the pathogen. All right. Figure 6.3 shows some human teeth that require dental treatment. D1. Identify the type of teeth in figure 6.3. Well, those are the ones at the front of your mouth called incisors. All right. 2. Explain how bacteria dissolve enamel to cause tooth decay. This is an explain question. So first of all, the bacteria use a sugar that's left on teeth as food for respiration. So bacteria use sugar left on teeth as a food source for respiration. And then when they respire, if they respire aerobically, they produce carbon dioxide. If they respire anaerobic, they produce lactic acid. Both of these are acidic. Carbon dioxide is acidic when it's dissolved in water, so you have lots of water in your saliva. And the acid dissolves the enamel. So this produces acid that dissolves enamel, and the acid is either carbon dioxide or lactic acid. They're both acidic. E. Describe 2 ways of preventing tooth decay. Well, one of the best ways to prevent tooth decay is to brush and floss your teeth regularly. Brush and floss teeth regularly. And another thing you can do is to avoid sugary foods. Avoid sugary foods. If you don't eat the sugary foods, the bacteria don't have the sugars to feed off of. Another thing you can do, I'm only going to write down 2 of them here, but other things you can do, you can visit the dentist regularly, and the dentist can make sure, give you steps to take if they are starting to decay. And fluoride in either toothpaste or water is also shown to prevent tooth decay. Now, if you look at some of the past papers from before 2016 for similar sorts of questions, you might notice that one of the answers is generally eat a hard, crunchy food like an apple. That does not seem to be on the mark scheme anymore, and the reason why that is, I would assume, is because apples contain a significant amount of sugar, so eating something sugary is not necessarily a great way to prevent tooth decay. Okay, so those are the ones that they want to hear now. It should be okay. All right, and now we have reached the end of this exam. I hope you found it useful and you'll do better on your exams. If you do like it, please press the like button, and we'd really appreciate it if you'd subscribe if you haven't already subscribed. And we love to hear your comments in the discussion section below, and have a great day.